Neuroscientists at Johns Hopkins Medicine have made a groundbreaking discovery by reactivating a specific memory circuit in mice, prompting them to seek shelter even when none is present. This study, published in Nature Neuroscience, sheds light on how memories are structured in the mammalian brain. The findings hold promise for developing new methods to study and treat memory loss in humans, particularly in conditions like Alzheimer's disease. By understanding these memory circuits, researchers aim to uncover the mechanisms behind memory formation and retrieval, potentially paving the way for innovative therapeutic strategies. The research team focused on two key areas of the mouse brain: the nucleus accumbens, known as the brain's pleasure center, and the dorsal periaqueductal gray, or dPAG, which is involved in defensive behavior. By stimulating neurons in these regions, they successfully reactivated a spatial memory, causing the mice to seek shelter. When we artificially reactivate those memory circuits in the brain, it triggers the mouse to do the same thing it did naturally, said Hyungbei Kwon, PhD, the study's senior author. This discovery highlights the intricate relationship between memory circuits and behavior. In the experiment, mice were allowed to explore a box with a shelter in one corner, surrounded by visual cues like triangles, circles, and stripes. These cues helped the mice form a spatial memory of the shelter's location. After acclimating to the environment, the researchers introduced a visual or auditory looming signal, prompting the mice to seek shelter. This process established a memory of the shelter's location relative to the visual cues. The team then used this setup to test whether reactivating specific neurons could trigger the shelter-seeking behavior without the presence of an actual threat. To achieve this, the researchers employed a light-activated gene expression system called CalLight, developed by Kwon in 2017. This system allowed them to selectively tag and reactivate neurons associated with the shelter memory. By activating neurons in both the nucleus accumbens and the dPAG, the mice sought out the shelter area, even when neither the threat nor the shelter was present. Interestingly, activating neurons in the nucleus accumbens alone did not prompt shelter-seeking behavior, while activating neurons in the dPAG led to random reactions. This finding underscores the importance of coordinated neural activity in memory retrieval. The implications of this research extend beyond understanding memory circuits in mice. Kwon suggests that these findings could lay the groundwork for reactivating or engineering memory circuits in individuals with Alzheimer's disease. If we understand the macro-level structure of memory, then we may be able to develop more effective strategies to prevent or slow down neurodegenerative diseases using this method, he explains. By mapping memory on a cellular level, scientists hope to devise interventions that can preserve or restore cognitive functions in patients suffering from memory-related disorders. Looking ahead, the researchers aim to explore the broader memory structure within the brain by selectively tagging and reactivating neurons with different functions. This approach could reveal how various memory circuits interact to produce specific behaviors. Understanding how all of these memory circuits work together will help us understand brain function better, Kwon emphasizes. The study involved collaboration with experts from institutions like the Max Planck Florida Institute for Neuroscience and the Zuckerman Mind-Brain Behavior Institute. With continued research, the team hopes to unlock new insights into memory and cognition, ultimately benefiting those affected by neurodegenerative diseases.